Up next, we will have Dr. Arulkumar Murugesan to present an article, Fissile Green Synthesis and its Potential to Anti-Cancer Activities. Please, sir.
Respected Director and most respected Chief Scientist, Baba Atomic Research Center, and our Chief Guest from Nepal, and other winners of the ISN Awards. Good evening to one and all here. And I thank to the organizing committee, organize a wonderful manner. So I am very happy to present my research findings, which is impact to the current society. So what is our research topic is the nanoparticle, how it can be react to the cancer cells. It is our major, in fact, so I don't want to go any other detail of the introduction and all other things. I just explain what we are going to work. That's enough for this time, I think. Okay, thank you. So I continue my research. Yes, these are the introduction. What is my background of the studies? The medicinal plants, how it is going to produce nanoparticles. So we know that different types of nanoparticle. It is like silver and gold and other metallic nanoparticles. Why our research is concentrating to this means our today we had many environmental issues which is leading to our research end of the objectives. So that's why we are focusing the green manner, how their green technology they are used to this inhibit and then reduce the pollution, as well as we want to life cycle development in our current scenario. So this what we are concentrating the medicinal plant and green approach. It is nanoparticle, it is produced from medicinal plants and it is combined together and found act and inhibit the cancer cells, which is are going to use our research. This is our objectives. I don't want to go there. Okay, so these are our results. Yes, a little bit explain what we are conclude that. So this is our result, main importance, which is apoptosis changes, which means cancer cell death. We focus that is control cells, it is a normal cells, which means there is no effect of the cancer cells. So look at here, treatment, the color will be, the lot of colors will be appeared. It is mean by cell death occurred in the nanoparticle. When we are giving our synthesized nanoparticle, which means produced from the medicinal plants, the nanoparticle, it is affect to the cancer cells. So how we are conclude that our nanoparticle, it is not affected to the normal cells. How it mean by, we are already checked the normal cells, which means we are isolated from normal human cells. It is not affected to the nanoparticle, and we are giving that cancer cells, they are inhibit and then they are reduced the cancer cells. So this is the lung cancer. So this picture was expressed as a lung cancer. So we have the number of the cancers. So we know that the different kinds of cancer will be increased day by day. So our aim to reduce this medicinal plant, then control the different type of cancer. This is our initial stage. We want to develop our initial stage and we want to develop the next future. And thank you. And thank you to my research team. This is my publication. And then future direction it is called. And my acknowledgement, where I studied in PhD from the University of Madras, and where I studied, continue my research in Bharathidasan University here, Tiruchirappalli. It is located in Tamil Nadu. And also, I thank to our research team, Dr. Anand JV, University of Madras, and Dr. Bala Shanmugam Paneerselvam, Central Leather Research Institute, Chennai. And also I thank to Dr. Rajendran Murugan, University of Madras. And also I great thank to Professor Dr. Devasena, Anna University, Chennai. And thank you. Thank you, Nanda.
Thank you very much, sir, for presenting your detailed study of your research work. Any queries from the guests and dignitaries?
Yes, sir.
I am Dr. Balakrishnan from Bangalore. It's a nice work. I just want to understand that you're working on nanomaterials. How you are introducing your nanoparticles of the medicine into the patients. It is through targeting the cancer cells actually or directly through medicines.
Sir, it is first we are initiate that in vitro studies. After that, maybe in future we want to trial to the in vivo studies. After only we are give to the human.
So still it's not targeted.
It is
to the
not
cells.
targeting. It is a preliminary experiment. We are starting our research.
So how can you find the dead cells and the real cells? How your chemicals is working?
Yeah, it is based on characterization and based on the experimental studies. I skip all the experiment here because of here we want to explain everything, and this is my presentation, which means where we are published. This lot of information is available.
Okay. Good.
Yes, sir.
Good work.
Can you name some planets which are the main basic for the cancer?
Yeah, Chlorissa super.
plants.
You are asking plants, yes, sir.
Yes,
Medicinal
the
plants.
medicinal
Yeah,
plants.
lot of plants are available, but we are target based on botanical classification. So we are target that is aromatic medicinal plants. They have potential inhibit to the cancer cells. Aromatic medicinal plants. It is belong to Solanaceae, and also it is Euphorbiaceae, and few are more Nimbiaceae. Like that, we are categorized the different type of medicinal
It's
plants.
aromatic means having breath or intaking medicines or
So intact medicines.
intaking medicines. But it's not concluded. Whatever your research is going on.
Yeah, it's are going, and also many of the research institutes they are conducting nano medicine, and we are initiated like Indian Institute of Science Bangalore. They are formulated drug delivery system. It is wonderful research. If possible, you can go read in the particular department.
What's your qualification, sir?
Sorry, sir.
What is your qualification?
Yeah, I am finished PhD from Madras University. Now I am working as
No, PhD in what? The subject?
mycology.
Mytology.
Mycology.
Mycology.
Mycology. It is based on the fungus. Yes.
Okay, okay.
Yeah.
Actually, it is already there in a Charaka Samhita. There are some plants are there to
Yeah,
cure
yes,
the
sir.
cancer.
Lot of medicines and
Medicines are there.
Yeah, yes, sir. We are
Means,
formulating and again we are want to reduce to that environmental issues. So when we go for the physical and chemical method, we are have the lot of side effects. So that's why we are manipulate the drug by using medicinal plants. It is a initial stage, and also we have lot of medicines are available based on the medicinal plants.
I wish you all the best.
Thank you, sir.
And we'll clap him and thanks to him.
Yeah, yes, sir.
Hello, sir. I'm Oli Katari from Naipa Mohali. I'm currently doing PhD. Sir, I had one query. Like you said, you are preparing a nanoparticle, right, out of this medicinal product.
Sorry, madam.
You are preparing a nanoparticle, right?
Yeah, yes, madam.
Can
Yes.
you please tell me what kind of nanoparticle you are preparing?
Silver nanoparticle.
So, are you aware of the toxicities that these nanoparticles cause?
Oh, yes. I skipped that, but if you're interesting, I will explain. Yes. So look at this first diagram. It is a normal cell line. So when compared to that normal cells as well as cancer cells, then only we conclude that it is not affected, this is not affected. Because when we go for the cancer cells, when we are taking that lung cancer cells, this is the normal cells. It is normal cells, it is going to be affect our silver nanoparticle. When we go for that A549 cell line, it is affected 100%, not 100%, 98 point something. These are when then we are concluding the normal cells will not affected to that green manipulated nanoparticle. It is only affected the cancer cells.
Okay, so you have done till 100 microgram per ml.
Yeah, yes, madam. Yeah, here. That is a MUG, microgram per
So
milliliter.
at such a high concentration only it is able to provide 100% cytotoxicity.
Yes.
Okay, and what was the particle size of your formulation?
Yeah, it is 20 nanometer to 15 nanometer.
And what is the route of the administration you are concerned?
Yeah, yes, madam. It is only preliminary cell line. We won't give the in vivo studies. It is only preliminary in vitro studies.
So because your particle size would determine, like your route of administration would determine the particle size as well.
Yeah, yes, madam. We can conclude that particle size. It is around a spherical shape, and also it is have the 20 to 50 nanometer size. We are
Because-
given in publications
Because
also.
if you are going to give orally in case, okay, so
yeah, yes.
any particle size below 30, I don't think it would be
Oh.
effective. Yeah.
Yes, madam, good question.
So-
But actually, you first understand, we human cells 20 to 50 micrometer size. When compared to that nanoparticle, it is a 10,000 times smaller. So it can be easily delivered to that.
It's not about that, sir. You please refer some good papers, because particle size is an important factor. If we give less than 30, it will just get washed out by the renal system. And so you please refer such papers so that
Okay.
it
Yes,
would be
yes.
helpful for you when you further go for in vivo studies.
Yes, madam, but we can understand what I mean. We give that based on the molecular weight and based on the charge, the drug will be released to the human cells.
But
So,
like based on molecular weight of the drug, right?
yes,
But you are putting it into a nano carrier.
sorry, madam.
You are incorporating that drug in a nano carrier, right?
Yeah, yes, nano, okay.
So the molecular weight won't matter in that case, because it is already entrapped in a nano carrier. So what will matter is a particle size charge, and particle size charge, and the PDI also, which we like.
Madam, many researchers, they are focusing drug delivery and directly they are give. Our research only direct in vitro studies. This is our initial
So
stage.
I understand that
Yeah.
this is a good work, but in case someone refers your paper and further proceeds for in vivo studies, these things will matter.
Yeah, yes, madam. We give the lot of information here.
Fine.
Yes.
Okay. And can you please tell me what is the method of preparation of your nanoparticle?
Yes, normally we are concentrating that cold extraction method.
It's a cold extraction.
Yes.
And you are using anything additional for reducing the particle size?
Yeah, silver nitrate.
For reducing the particle size.
Which particle
Like
size?
you are preparing with cold extraction method. By that method only you are getting the particle size of 20 to 50?
Yes, madam. I will explain the procedure. We take the plant extract. Okay. After that, we are adding that silver nitrate. Then it is formulated after 24 hours. We can conclude that based on the UV spectrum. Then we go for the other characterization process like FTIR, SEM, and other instrumental analysis.
So you
Then
are
we conclude that this is a synthesized nanoparticle from medicinal plants. Yeah, yes, this is the experimental.
so you are checking the particle size only at in SEM.
After formation of the 24 hours only we want to check. Yes,
In SEM. And
yes,
no
yes.
other instrument you
Yes,
are using?
no. We can go for the up to 3 months. They are without changes. You are asking that stability, I think.
No, I'm not asking about stability. Stability is a different perspective. What I'm asking is that you are saying 20 to 50 nanometer is the particle size of your nanoparticle, right?
Yes, madam. It is up
So
to.
how you are confirming that it is 20 to 50? Like how you are checking? I'm asking this.
Yes, based on the SEM, scanning electron microscope, and transmission electron microscope.
Okay.
Yeah.
Okay. Thank you.
Thank you, madam.
Sir, one more question. So 24 hours with the help of magnetic stirrer or homogenizer?
Yes,
Which one
yes,
you
sir.
have?
We are trying to magnetic stirrer and normal conditions also, but it is good on normal conditions also. Normal conditions only.
You said that 24 hours you are stirring.
Yes.
So
No,
20
no. We are not stirring.
Okay.
We are just keep in dark condition, then we get the result.
So without any stirring, you
Without
are
any
able
stirring.
to get the nanometer.
No. And also we are trying to stirring condition. It is half of the strength, and we want to go for the stirring conditions because of our previous researcher in our own laboratory. They are doing the normal condition, so we are also with that reference we go and then we produce that nanoparticles.
Thank you.
Thank you very much, sir.
Thank you.
Thank you very much for the information also.